I'd like to start by thanking the organizers. I got to know that I'm going to be doing this just 2 days ago. I just got here yesterday. So forgive me for a kind of impromptu composition. Okay, so first of all, I must begin to thank my gurus, namely my students and postdocs. And it's again, it's more like an orchestra. We have a few people doing theory, analytical theory, simulations, of course, and then the hard work of doing experimentation is done by several people, and some people do both experiments and theory. I must be very, very grateful to these people. Without that, my talk would be empty. Okay, so what's the context? The context is this. Nowadays, there is a huge interest in what are called membraneless organelles. These form spontaneously inside a cell, both inside the nucleus and outside the nucleus. And these organelles do not have a membrane. However, they have fantastic, very unique biological functions. How come those functions come about without the pumps like sodium pump, potassium pump, without that, no membrane, no channels? How do they survive? How do they do these things? More importantly, how do they even organize themselves? And they are made up of what are called intrinsically disordered proteins. These are not easily foldable. These are not like, evolution point of view. We always pay attention to foldability of a protein sequences, but here more or less they are kind of schizophrenic. They are more like a charged macromolecules, polyelectrolytes, the same thing protein-like. So that's why we call them intrinsically disordered proteins, and they themselves can organize, but quite often they organize in the presence of RNA or polyphosphoric acid. That's one of the ancient polymers in our evolutionary history. All right, and of course the small inorganic ions, basically magnesium, sodium, potassium, sometimes calcium, etc., etc. Now, when we look at them, they have hierarchical structures. They undergo order disorder transitions. First they are born into some sort of a droplet, and then as a function of time, they become bigger and bigger. And again, as a function of time, here number of days, for example, they convert themselves into some solid-like structures. So first they are like a liquid-like, more like oil in water, and then eventually they become more solid-like. And then when you pay attention to the structure inside these solid-like hydrogels, there are lots of fibrillation, etc., etc. And these fibrillations and as well as the solid-like structures are implicated in several diseases. But apart from being relevant to diseases, even the functions, how the local chemical detail gets amplified into these larger scale structures is really a fantastic challenge for us. So say broad length scales and time scales. So like some of these objects when they are born, they are about 20 nanometers, and eventually they become 5 microns. And then in terms of time scales, several decades, right, of time, say 10 nanoseconds to days. And this is one of the grand challenges. Why? And if I put my pointer anywhere inside this, it's like a Coulomb soup. It's really charged, electrically charged, water, dielectric constant is not known. It's really a dielectric function. It depends upon the length scale and time scale. And the reason why this is dubbed as one of the grand challenges, in particular molecular biophysics, and to some extent polymer physics, is lots of forces operate at the same time, simultaneously. It's a confluence of electrostatics, monopoles, dipoles, quadrupoles, etc. And chain connectivity is a topologically correlated object, classical string, if you like. It's loaded with conformational entropy, and that entropy plays a magic in terms of making peace between various interactions in here. I need another long-range interaction of hydrodynamics. And the chemical sequence of these proteins is extremely crucial, and more importantly, water. That's an extremely important component here. And of course, charged polymer can never be alone. Maybe in theory it's possible to do, but never alone. It's always surrounded by counterions because electrically it has to be neutral, the system has to be neutral. So the counterion physics can be a very significant physics dominating what's going on here. And there is also hydrophobicity that is more like oil-like van der Waals type interactions. All these things are taking place at the same time. So what's the consequence of this? As I pointed out already, structure is hierarchical structure, dynamics is hierarchical, and the liquid-liquid phase separation, people say as soon as they see something like this, then very quickly we can jump into the conclusion that this is more like a two-component phase separation, liquid-liquid phase separation. Of course, the transport, the kinetics of the whole processes, these are all the key issues that one has to address. We cannot do them today, all these things, right? But we got to begin to look at them in a more systematic way. So, given that premise, I want to remind myself that if I look at the some aggregate like this is from our simulations, and if I put my pointer here, one of them is an IDP, intrinsically disordered protein, and if you look at the sequence of this, we have lots of things going on. There is a hydrophobic group, more oil-like, and then here is a lysine that's positively charged, and then here is aspartic acid that's negatively charged. We have been biased to look at the electrical forces alone, monopole interactions alone so far, but it turns out that there are other groups, like a neutral, so-called neutral groups, they are not silent, right? If you take neutral groups, here is an asparagine, which has a dipole moment of a 4 dBi, and then we have the glutamine, and that has a dipole moment of 5 dBi, and if contrasted that with our matrix of life, water, the dipole moment is 1.8 dBi. So compared to that, these are not insignificant. And we all know the magic world that water has created for all of us, right? And if that's the case with that dBi, that 1.8, and imagine all the things that can happen from these entities as well. So let's think qualitatively here. So if I take a sequence of a polymer here, then I have, let's say this is a charge-charge interaction, and the strength of the Coulomb interactions, we write it as a Beerem length, which is 1 over temperature times epsilon. Epsilon is a dielectric constant, but that itself is temperature dependent because of a solvent reorganization. And then we have, that's the strength of the interaction. Now we have charge-charge interaction, which could be attractive or negative, attractive or repulsive, depending upon the sign of the charges. And then we have, charge dipole interaction, that's 1 over r squared, attractive, dipole interaction 1 over r cubed, that's attractive. So we have attraction, repulsion, they're all in a sequence. And then the hydrophobicity, van der Waals interactions, is the tradition in this field is to write in terms of this parameter, which is called Flory-Huggins chi parameter, and that kind of captures, parameterizes the van der Waals interaction. So we have all these things going on here: charge repulsion, charge dipole attraction, and therefore what we have is, depending upon the sequence, we already have a frustrated electrostatical system right away. When we write the sequence, it is not a simple polyelectrolyte, it is not a simple dull polyethylene-like polymer, right? It is a highly frustrated right away at physiological temperatures, physiological conditions. So therefore, that falls into our category of trying to understand in a proper fashion. And the piece between this frustration is kind of conducted by the conformational entropy, which is connecting all these things together. Of course, indeed.
From the images that people show, it's clear that you do have a the phase separation is clean, right? That there is a surface tension, you have spherulites. How much is known about the chemical, the composition difference? What are the two phases? Is that clearly established?
Not in this community. In our traditional polymer, your field, my field, the past, we know what a liquid-liquid phase separation is. It is because it's simply a two-component system, and we know how to measure the compositions of the coexisting phases. We can predict them, on and on. But in here, that is not established. Very quickly, because it looks like our traditional liquid-liquid phase separation, the biologists have been calling this to be liquid-liquid phase separation. It is not yet well understood, and that's in fact my point, because it's a multi-component system, for example. But still, there are opportunities to make some progress, right? Okay, thank you. So, again, let me remind for some of the younger colleagues in here, okay, normally when you talk about electrical potential away from charge source, here it's a charge macromolecule, however way it's decorated. We have been paying attention to only monopoles, it's total charge. That's the way we have been doing it, because that's 1 over r is much stronger than 1 over r cubed in terms of interaction strength. So that's our mind has been kind of influenced by that one. That already is very complicated. It's more like a plasma. We have the Coulomb interactions, the screen Coulomb or Yukawa, right? Kappa is the inverse Debye length, which is influenced by ionic strength, salt concentration. But now when we look at the dipolar interactions, like the P is a dipole moment. Although it's 1 over r cubed, the dipole moment could be very large. Particularly for these molecules, the dipole moment is not a very small number. I gave you some examples before, particularly when you put them together, the dipole moment could be very large. That means that the interaction strength, like interaction strength between dipole-dipole, although it goes like only 1 over r cubed, in terms of range it's weaker than the Coulomb, but on the other hand, the P square is very large. And this is not new. People knew this all along. Suppose they were to be randomly oriented. If you do that, then it becomes 1 over, it will become 1 over r to the 6. And this is what we call Keystone forces, which is responsible for gas being liquefied at a low temperature, right? It goes like P to the 4. And this is the origin of van der Waals interactions. We all know that. Yeah, please.
Remind me, we have the dipole-induced dipole, and then the third one, the London type.
Right.
Keystone is dipole-induced dipole?
No, it's the dipole-dipole, but randomly oriented. Okay. Right. But if you do the induced dipole, you still will get 1 over r to the 6. That's Debye, for example. And then London forces. They're all in the same class. They are all grouped together as van der Waals. Okay, so if that's the case, even with two simple molecules, imagine what will happen with the polymers. When you put all these things together, this effect is going to be amplified. The Keystone force is going to amplify, and therefore it's important. Okay, so given that, let's think a little bit more. I'm not going to be technical. I'm not going to derive lots of equations from here. So let's think qualitatively here. If the temperature is very high, then we can call, in our language, we can call this to be anneal, because that's our Keystone 1 over r to the 6, because they are randomly oriented, although they are tethered to the backbone of the polymer chain. But on the other hand, if you lower the temperature, they are not randomly oriented. Then it belongs to quasi-quench or quench system, where the orientations are kind of more or less fixed. They are not randomly oriented, right? So one has to make a transition from this to this when we do the experiments, to go very carefully look at when I could treat this problem to be annealed, when this problem should be quenched. Well, the technology to do all these things, all these effects is available to us based upon field theory. Write the Edwards Hamiltonian we call Feynman path integral. You write the sequence in here, put any interaction that we think of. I already gave you expressions for the interactions, put them all in here and work out. We know, we just do the field theory, right? Perturbative theory, variation calculation, self-consistent field theory, vertex corrections, all those things can be done. So I'm not going to do that here, okay? It's doable. Okay, now let's go to the problem. Let me, in the interest of time, let me show one petite result here. Let me consider a collection of, okay, to make this, to illustrate the technique, let me think of a polymer which is made up of only dipoles, right? One monomer carries a dipole, another monomer, next monomer carries a dipole. They are tethered together. There are n number of chains in the system, and capital N is the number of dipoles per chain connected contiguously, and then F is a fraction of dipoles which are going to have the correct orientation for the quenched case, okay? And then out of all these possible dipoles in a chain and many chains, only a certain fraction of them are going to be participating in the quadrupoles. Let's say Q is a fraction of those dipoles in the quadrupoles. That means by construction, let's assume there are going to be NC crosslinks, physical junctions. They are not permanently crosslinks, physical junctions, which is given in terms of these numbers, the Q, F, and N. There will be 2 to avoid double counting. Then we can write the free energy. Yes, and the combinatories of how we are going to assemble this aggregated, this network structure, that is this one, that depends on number of crosslinks. And then of course the epsilon is the quadrupole energy e to the epsilon times the number of those junctions by kT. Then I'm not writing an explicit formula, but that can be found in here. So you minimize this free energy with respect to number of crosslinks, and you get a very simple, beautiful formula here. Crosslink density goes like volume fraction of this polymer 1 divided by 32 pi square. 1 by 32 pi square is a small number, and then there is e to the epsilon over kT. So the crosslink density turns out to be small, and that's the clue. That allows us to do some nice analytical calculations. So let's do that. So given this, if there are NC crosslinks in our field, we know how to look for free energy of mixing, and then elasticity, because between junctions it acts like an elastic wire. It's because it's more like a hydrogel, and all the interactions in the system, monopole, 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 dipole, all those interactions depending on the sequence. And based upon this, we can calculate the distribution function for the number of aggregates with m number of polymer chains in that aggregate. So depending upon the polymer protein concentration, salt concentration, and temperature, one can easily predict the size of these aggregates, and also the most probable size of an aggregate under certain conditions, how many number of chains there are going to be. All those things that can be easily calculated. Again, the details are in here. So as a consequence of this, using a StatMech, and in the dilute limit, we have isolated chains, and they can associate, dissociate, they can collapse. When you add salt, the dipole-dipole interaction will be screened, it will open up, and then they begin to make aggregates after certain critical aggregation concentration, and that depends upon whether temperature is a variable or pH is a variable, salt concentration is a variable. Depending on which is the experimental handle, we can predict the sizes of them. And then we increase the concentration of this, polymer protein concentration of this, eventually we get the percolation line and then a gel, and there's a tricritical point in here, gelation line comes and hits with this one, and that's a critical temperature. And the fluctuations are not easy here because the fluctuations involve not like a usual liquid-liquid, but there is also what we call microphase separation in this particular mesomorphic state. And then the critical exponents are not icing-like. There are new exponents that are coming about, and when you go to temperature below this, we have liquid-liquid phase separation. The two phases are coexisting, Shrikant. Two phases are coexisting, but each phase is not the usual isotopic case, but then inside this you have microphase separation. Okay. And then when you go below a tricritical point, we have a liquid gel phase separation. Gel is coexisting with the liquid, and then that liquid is not a simple liquid, it's a structured liquid with a hierarchy of structures. Okay, given this, now I like to go to the dynamics part, because that's the title of my presentation, today's presentation. And that let's think about this. Before I talk about dynamics, just for the benefit of younger colleagues, let me summarize what we know in polymer physics developed over 7 decades. In a dilute solution, you put a polymer chain with the monomer length is, let's say, this little L. Capital N is the number of monomers in this chain. It has, we heard very nice trajectories of random walking objects. Here is a trajectory of an electron in time, which is mapped into a conformation of a polymer chain, and this is the end-to-end distance vector. But this chain is embedded in a hydrodynamic liquid with viscosity eta naught. This monomer is connected with, correlated with this monomer with the hydrodynamic interaction, and this is connected with that one, this is connected with that one, on and on and on. There is big orchestra is going on, hydrodynamic coupling. And for this particular case, paper and pencil calculations can be done to show the end-to-end distance vector. The correlation function is, of course, the equilibrium is a Gaussian chain, number of steps times step length square, and then e to the minus t over tau z. Tau z is a characteristic time, which is written by Bruno Zim, and it turns out it was also done by Raman. Okay, so that tau z goes like chain length, number of monomers per 3/2. Okay, and this physics is dominated by viscosity here. If you increase the protein concentration or polymer concentration, the chains would interpenetrate. When they interpenetrate, the hydrodynamic interaction is is screened, just like a Coulomb interaction is screened into a Yukawa potential similar to that. Here hydrodynamic interaction is screened by interpenetration. Now the correlation function of the end-to-end distance vector goes like again NL square for Gaussian chains, e to the minus t by different characteristic time. That characteristic time is given as n square. But the important thing is here the viscosity doesn't appear because hydrodynamics is screened. Now it's a friction of every monomer that matters. Okay, that's the quick summary of polymer dynamics. You know, books are written on that one, right? Now let me consider our gel, hydrogel. In general, if I take a gel like this, there is a junction, and between this junction there are m monomers in a strand. Let me write the end-to-end distance vector of this strand between two junctions. That's a PMT. M is number of monomers at time t. The way we interrogate gels, understand the dynamics, either elasticity or a local dynamics of the monomers, is using light scattering and neutron scattering, usually light scattering because the time and length scales are appropriate for hydrogels. So in light scattering, what we do is this: we have an incident beam with an incident wavelength lambda, and there is a scattering angle theta. We measure the scattering intensity at a certain scattering wave vector q, which is the reciprocal of lambda and sine theta by 2. Of course, using the placement of the goniometer, you can choose the angle theta, again depending upon the particular incident beam. And the theory is very well developed. Toyo Tanaka in 70s, and later on several people did this, right? So from there, you measure the scattering intensity, and then the theory tells us electric field correlation function, electric field at q, scattering vector at q at time t, dotted with the electric field at q at a 0 time. That correlation function is given by G1. G1 is related to the intensity correlation function. Experiments will give me the left hand side, and on the right hand side, this the theory shows is basically the correlation function that I wrote down before. And as I showed you before, it turns out to be an exponential decay. This is a standard theory of what people would do. But now when we have the hierarchical structures, I would expect a strong deviation from this. Okay, that's the idea. So given this general trend that is very well established in the literature, now we are going to move further. The way we do this is the following. Okay, suppose I look at a formation of quadrupole, and then two dipoles are going to come close and make the pair, and they have to cross a barrier, epsilon A, and then this is gain in energy, quadrupole energy. So that's the pathway by which it is going to make an association. So the rate of association is given in terms of this epsilon A. Now the rate of dissociation is given by this barrier. So that's going to be epsilon plus epsilon A. So that's the kinetic equation. Now let's look at the formation of the network. I already told you that the cross-link density is very small. If the cross-link density is very small, it's a Poisson process, right? Low crossing density. So in a Poisson process, the distance between the number of crosslinks is going to be, which is m, is going to be exponentially distributed. So probability of a strand of a certain length m is going to be that Ka that I wrote down here, e to the minus Ka m. It's exponential. Exponential is a very friendly quantity, right? So that means when we look at the dynamics, the m that I told you is distributed this way. For each value of m, there is a relaxation time. And that's how the hierarchical dynamics comes about, right? For each m, there is a relaxation time. Then I have superposition of all those things. And let's do that. So I need to know what the relaxation time for m monomer is. Now, when I have a chain length n, in a hydrogel now, hydrodynamics is screened because it's really a concentrated system. Therefore, I need to look at the friction coefficient of a particular monomer that's going to be binding with another dipole to make a quadrupole. That I can write in terms of friction coefficient. Friction coefficient zeta c, which is participating in the cross link, would be the bare native friction coefficient times this quantity, which is related to the chemistry, the barrier as well as gain in quadrupole energy. So now, according to Rao's dynamics, the tau Rao's is proportional to, this is the pth mode, it's resolved into Debye modes, and in a polymer field they are called the Rao's modes, but they are really Debye modes. So when you look at the tau Rao's for the pth mode, that will go like n over p square. Now what I'm interested in for the m number of monomers in the strand, I'm interested in the n over mth mode, right? So therefore the Rao's time for the n over mth mode would go like m square. and the prefactor is specific to the particular system. That is the non-universal, but this one is m square. So therefore, for PMT, that correlation function will e to the minus t. This is the relaxation time, Rouse relaxation for m. That goes like m square here, and AR is this number. That's chemistry. Okay, given that, let's do the calculation. All right. So G1T, which we measure experimentally, is given by this blue term is a Rouse dynamics for m strand. Yeah, in the strand with the m monomers, that's 1 over t over m square. And the probability of getting that m is given by the Poisson process, that is e to the minus Kam. We have to do the saddle point. What we get is, do the saddle point, you can do it in your head. G1T goes to e to the minus t over some relaxation time, characteristic time, which is specific to the chemistry. Power and exponent beta, beta is exactly 1/3. That's universal, 1/3. And this is not universal, that's depending on the chemistry. Now the question is, okay, can we do experimental validation of it, this prediction? We can't. We talk to the chemistry. Why we can't do that is the molecules are not there. You cannot make lots of, 1 gram of a polysubtrione. It's not very easy to, particularly pure PC. It's not possible to do. We have now learned how to make them. So we went and looked at other experiments in our lab. So here is polyacrylamide gel, 1% chemical crosslink. We do the light scattering experiment, and that's the correlation function we measured. And you can, I don't know whether you can see it. There is also a red curve on top of this the black data points, and that's the fitting. And the difference between that and this is given by these blue dots. There's a single exponential, e to the minus e. This is a chemically cross-linked system. There are no handshaking at all. On the other hand, if you suppress the chemical cross-linking, but let the polyacrylamide to physically associate and dissociate, then we get, this is the gel mode. From there we get the elastic properties, modulus, bulk modulus and shear modulus, and then from this fitting point you get beta square 1/3. Okay, so that's for that system. Now let's do another system. So here is a polyacylate gels, polycations, polyanions put together, neutral overall, and again beta square 1/3. Another example, hydrophobic group. This is we took a lysozyme, denatured lysozyme, and it's able to fibrilize. And then those fibrillized regions could participate in hydrophobic physical crosslink, and again beta square 1/3. So let's try to put them all together, right? By adjusting the characteristic time, because that is specific to the particular system, always you get beta equal 1/3. So there is a universality in terms of hierarchical dynamics, but the characteristic time is specific to the system. So this gives us an ability to tune the clock, tune what kind of time scale where this particular object would express itself in terms of functionalities. On the other hand, the power law is 1/3. This is water, room temperature, 96% water, 98% water, but it looks like, it pretends like a glass, the traditional low temperature inorganic glass, right? But in terms of structure, StatMech, it is, it's very nice. Okay, so with that, I think I have enough time to conclude. Universal is the universal hierarchical dynamics associated with hierarchical structures in a diverse condensate systems. Almost all, as far as we know, we have tried another 10 systems, they all show beta equal 1/3. Okay, diverse systems, we have the universality. This recipe, this theory allows us to easily detect physical gels from chemical gels. You do one experiment, light scattering, put it in, light scattering experiment, you figure beta equal 1/3, then it's a physical gel. If it's a chemical gel, no. And also, quantitatively we can determine a physical crosslink density, which is not known so far. We can do that. More importantly, we can tune the dynamics of hierarchy in a desired way, in terms of designing and also understand what's going on. With that, I'd like to thank you for your attention.
So the 1/3 here is coming from the Poisson nature of the crosslink positions and Rouse. Yeah?
Yeah.
Okay. Now, in terms of discussing.
So for in general, in view of all the beautiful talks we heard, you want to dissect the process into parts and allow them to express those parts. If you do that, you get the hierarchical structure. And when you separate them, you can separate them in so many ways, but Poisson is usually nature's choice.
Okay.
Sorry.
Just to add on this question, so instead of Poisson, suppose I take a power law distribution for the segments.
I did not do that.
But
Yeah,
like
you could do that. You could do that. When you do this, the preliminary calculations show that there will be logarithmic corrections. Right, because if you do the saddle point, you can do it in your head. Logarithmic correction. That is not going to overwhelm this beta exponent.
okay.
I think so. Yeah.
Do you have any simple argument to derive the beta equal 1/3?
I just did.
Yeah, but I missed my sense part.
Okay, sure. Let's do that together. Let's do it together. All right. So here, simple way, right? Here you have m, the 1 over m square. Okay. So take the derivative of this, and then take the derivative of that. That will be m cubed minus m cubed, right? M cubed will go like t. So m will go like t to the 1/3. Put m square, t to the 2/3, t over t to the t to the 1/3, and m is what? T to the 1/3, t to the 1/3. Saddle point, just, or count the powers. I cheat, right? So what I do is, here, like this, bring this here, m cubed goes like t, so m is t to the 1/3. Sorry.
Questions? We have plenty of time.
It's your time to joke.
I have a joke. Now I understand why people think I'm a theorist.
Yeah, go ahead.
Yeah, so in terms of how this might apply to other cases where you have, there's a particular combination of Rouse dynamics with a particular form of polydispersity, right? So even if we go with Poisson being typical somehow, the Rouse dynamics will not be, right? So, but could there be some robustness to this result?
All right,
Goes
so look.
beyond-
Let's take Satyen's problem, right? Rouse goes like what? The time, relaxation time goes like m square. Einstein's theorem tells us relaxation time, Einstein's theorem tells us relaxation time goes like length square, right? That's how n square comes in. Any phenomenon, natural phenomenon, which would obey Einsteinian dynamics, Einsteinian behavior, would give me what I call Rouse in this system. That's the generality. And then any Poisson process in conjunction with that one will be this.
But in some cases, you might have some conservation laws where modern E-type of dynamics comes in.
That's absolutely right. That's absolutely right. Yeah. Then it would be of a different hierarchical structure. That's what we see in spin glasses, for example.
So, Muthu, is this independent whether you are like a physically associated system?
I mean, you can be above or below the percolation threshold.
Exactly.
So does it matter?
No, it doesn't matter.
So you're saying it's universal?
It's universal. Either in the aggregated structure, inside the aggregate microphase separate aggregated structure, then as long as they are physically dancing around, making and breaking, this is true. And inside the gel, it's true.
So if you do a rheology experiment, a plateau will be just separation.
Absolutely right. That's what we are doing right now. Good.
Now, dynamics, you say so the cheap dynamics. Cheap dynamics may be fun.
If I do Zim dynamics, right, we can do the same calculation here. It will be, instead of 1/3, it will be something like 0.34, 0.36, I guess.
I mean, the deviation
Very, very small, very small. There is an experiment that is being conducted in collaboration with the neutron scattering people, because there the concentration doesn't have to be large. We can interrogate even within one intensely disordered protein. And then it's hydrodynamically loaded. Then we are looking at the incoherent structure factor, coherent structure factor, we are going to. There, the exponent would not be 1/3 precisely, it will be a little bit higher. Yeah, but it will be much less than half. Right, if you do incoherent neutron scattering, for Rouse dynamics, it will be exponent should be something like half, and for ZIM, it will be 2/3. This is way below because of the hierarchy.